Usually when we talk about any kind of medical implant, there is this unspoken expectation of structural surrender.
Right. Yeah. Like the body just has to deal with it.
Exactly. Like if you put a titanium joint in your leg, to replace a shattered hip or something, the bone just has to accept it.
It's an act of engineering dominance, basically.
Yeah, you bolt the metal in, bone heals around it, and the biology just sort of submits to the geometry of the machine. It's a purely mechanical exchange.
And we are honestly very comfortable with that paradigm. I mean, we like things to be visible.
Totally. We want it to just snap into place.
Right. To be categorized, to snap in and stay there.
Yeah.
We sort of approach the human body like like a biological car engine.
But the moment you step into the world of neurology, into the soft, pulsing, microscopic realm of the brain, that whole expectation of mechanical surrender is completely
Oh,
broken.
it shatters completely.
Because we are currently looking at a technological landscape that isn't really a hospital. It's a battlefield.
That's a great way to put it.
And the fatal assumption driving literally billions of dollars of research right now is that if we just, make our materials softer,
Uh,
or if we just make our wires small enough, the brain is eventually going to accept them.
which is just not true.
No. But we're talking about almost every brain-computer interface you've read about in the news. The Utah arrays that look like tiny little beds of nails.
Yeah, or the Neuralink threads that the robots sew in.
Right. The advanced flexible polymer grids, all of them. Every single one is trying to force a surrender.
Which is a losing strategy, frankly.
So today's deep dive is all about why that strategy is failing and, more importantly, what the alternative looks like. We are looking at a paper by an independent researcher named Zachary David Enfield.
Yeah, the paper is titled Organic Quantum Interface.
And the foundational argument of this document is that our entire approach to neuroengineering isn't just slightly off, it is fundamentally, biologically false.
Exactly. His whole premise is that we've been trying to solve a biological immune response using material science.
And that right there is why long-term brain interfaces keep failing, right?
Precisely.
So that's our mission today. We are breaking down Enfield's wildly different architectural approach. We aren't looking at metal today.
No metal, no silicon, no carbon nanotubes.
None of it. We are examining a biological scaffold that is literally designed to speak the brain's native quantum language.
It's a massive shift in thinking.
It really is. And to understand this paradigm shift, we're going to break down this architecture through two core structural pillars from the paper.
Right. The six-layer design and the trimodal power system.
Exactly. The six-layer design moves from the absolute surface of the brain all the way to an external artificial intelligence.
And the power system allows this device to run endlessly, which is crazy, without a battery.
It's science fiction level stuff, but it's real biology. The best way I can frame this for you listening is, imagine trying to learn French by aggressively shoving an English dictionary at someone in Paris.
Just repeatedly hitting them with it.
Right. Just over and over until they understand you. That is essentially our current brain-computer interface technology.
It's brute force.
Today, we're exploring a device that actually bothers to learn the brain's mother tongue. But to understand why this new language is even necessary, we have to look at the crisis of wires.
The scarring.
Yeah. We need to talk about glial scarring. Let's paint a picture of what actually happens when you put a modern interface into a human brain.
Okay, so the moment you implant a synthetic probe, say it's a stiff tungsten electrode or even one of those flexible silicon threads into living neural tissue, you trigger an ancient, highly evolved defense mechanism.
The brain does not like intruders.
Not at all. The brain doesn't see a miracle of modern medicine. It sees a microscopic invader. So the central nervous system deploys its immune cells.
Primarily microglia and astrocytes, right?
Exactly. They swarm the implantation site. And they are reading the surface chemistry of that wire. Because it lacks the specific protein signatures of native tissue, they initiate this huge inflammatory cascade.
They basically treat it like a splinter.
They do. They build a wall around it,
A literal wall of scar tissue.
encapsulating the foreign object completely. And the crazy thing is, the entire neural engineering field has spent decades treating this as a failure of materials.
Right, the logic has always been, oh, our silicon is too stiff.
Yeah, they say the brain is the consistency of warm jello and we're sticking glass needles into it.
Yeah.
So what do? They try to coat the needles in these super soft hydrogels.
Polyethylene glycol dimethylacrylate, I think the paper mentioned.
Yeah, that's one of them. They are desperately trying to lower the mechanical stiffness, what engineers call the Young's modulus.
And the paper points out the sheer scale of that drop. Because, silicon is incredibly rigid. It has a stiffness of something like 200 gigapascals.
Which is basically rock hard.
Yeah, and they've engineered these hydrogels to bring that down to 15 kilopascals just to match the squishiness of the brain tissue.
Or they use flexible parylene C substrates, so the wire can sort of physically bend and sway with the pulsing to the cerebrospinal fluid.
They are bending over backward to make the object mechanically invisible.
But Enfield's premise just completely destroys that logic.
Yeah.
Because glial scarring is not a materials failure.
It's an immune success.
Exactly. The microglia don't just care about stiffness, they care about biological identity.
Right.
When they encapsulate that electrode, that chronic neuroinflammation produces continuous oxidative stress. It's a highly corrosive chemical environment.
It's literally eating the wire.
Literally.
Uh-huh.
It degrades the insulation and eats away at the active recording surfaces of the electrode.
So over months and years, the signal just slowly degrades.
Until the device is completely deaf.
Yeah.
Just walled off by the very body it's trying to read.
Wow. Okay, so the brain doesn't need a softer wire. It needs a conversation partner it actually recognizes.
A biological peer.
Right. Which brings us to the core of Enfield's paper. The aromatic alternative.
Yes. This is where it gets really fascinating.
If we aren't using silicon and we aren't using traditional flexible polymers, what is the alternative? To answer that, we have to zoom way, way in.
Past the cell wall.
Past the cell wall, into the quantum biology of the neuron itself.
We really have to unlearn the high school biology textbook version of a brain cell here.
Yeah, because the classical model treats the neuron like a simple biological wire.
Right. You have a buildup of ions, the membrane depolarizes, an electrochemical wave travels down the axon.
It squirts some neurotransmitters across the synaptic gap, and the next neuron fires. It's very mechanical.
Very classical physics. But that electrochemical action potential, the actual thing we've been trying to measure with our metal electrodes for decades, is actually just the macroscopic printout.
It's like the exhaust fumes of the actual thought.
That is a perfect way to describe it. It is the heavy, slow residue of a much faster microsecond-level computation happening deep inside the cell architecture.
And the hardware for that internal computation is the microtubule.
The microtubule, yes.
Now, microtubules, these are usually described just as the scaffolding of the cell, right? Like the internal skeleton that gives a neuron its shape.
Structurally, yes, they do that. They are cylindrical polymers made of a protein called tubulin. They form these tiny tubes about 25 nanometers in diameter.
So they're microscopic structural pipes.
Basically. But their function goes far beyond just holding the cell's shape. They possess the densest concentration of aromatic amino acids in the entire cell.
Okay, let's break that down for everyone. Aromatic amino acids. We're talking about
Yes.
specific molecular shapes, right?
Yes, rings of carbon atoms that share electrons.
Right.
Each tubulin dimer, which is just a single building block that clicks together to form the microtubule cylinder, contains exactly 86 aromatic rings.
86 rings in every single block.
Yep. If we look at the breakdown from the paper, that's 8 tryptophan, 36 tyrosine, and 42 phenylalanine residues.
That is a lot of rings.
It is. But what matters here isn't just the sheer number of these rings, but their precise geometric arrangement.
The spacing.
Exactly. These aromatic rings are spaced about 1 to 2 nanometers apart, center to center, suspended in the protein matrix.
Okay, a 1 to 2 nanometer spacing. Why does that specific distance matter so much?
Because that spacing is structurally analogous to the chromophore networks we see in photosynthetic proteins in plants.
Wait, like how a leaf absorbs sunlight?
Precisely. When a leaf absorbs sunlight, it achieves near-perfect energy transfer efficiency by utilizing quantum superposition.
So the energy isn't just bouncing around randomly.
No, it exists in multiple pathways simultaneously to find the absolute fastest route to the reaction center.
That's incredible.
And inside the microtubule, particularly among those 8 tryptophan residues, you have a massive opportunity for a similar phenomenon.
Because of the shape of tryptophan.
Yes, tryptophan contains an indole ring system. It's a fused double ring structure. And because of that shape, the electrons aren't tightly bound to a single atom.
They sort of float.
They become delocalized, forming this cloud of negative charge above and below the molecule.
And because these tryptophan rings are spaced so perfectly close together, those electron clouds start to, bleed into each other.
Exactly. The term in molecular chemistry is pi-p stacking. The pi electron orbitals of adjacent rings overlap.
Creating a pathway.
They create a continuous, highly conductive topological pathway. It forms a quantum coherent channel that operates at terahertz frequencies, running straight down the core of the microtubule.
Okay, so the theory Enfield is building on here, which is gaining immense traction in the field of quantum biology, is that the brain conducts its fastest, most fundamental information processing through these stacked aromatic rings.
Via quantum optical mechanisms, yes.
Okay, I have to push back here.
I figured you might.
Because, I mean, if you're picturing a quantum computer, you are probably thinking of massive, room-size rigs built by Google or IBM.
Right, the giant gold chandeliers.
Exactly. Suspended in a vacuum chamber, cooled down with liquid helium to a fraction of a degree above absolute zero.
To stop decoherence.
Yeah. They do all of that just to stop the fragile quantum states from collapsing the second a stray molecule bumps into them. So how can the human brain, which is a balmy 37 degrees Celsius, wet, chaotic, full of moving ions, possibly sustain quantum coherence?
It sounds impossible.
Doesn't the thermal noise just instantly obliterate the quantum state?
That exact argument was the primary club used to beat down the field of quantum biology for decades. Physicists looked at the brain and said, it's too hot and too messy. It can't happen.
Right. It seems obvious.
But the resolution to this paradox came from looking back at those photosynthetic plants. The mechanism nature uses is called phonon-assisted quantum transport.
Phonons, meaning units of vibrational energy or basically heat.
Heat at the molecular level is just vibration. In a synthetic, human-made quantum lab setup, heat destroys coherence because it introduces random, chaotic vibrations.
It shakes the delicate structure apart.
Exactly. But a biological protein is not a random environment. It is an incredibly highly structured, evolved matrix.
Oh, I see.
The thermal fluctuations, the specific ways the protein bends, twists, and vibrates at body temperature, they do not act as destructive noise.
So the protein isn't vibrating randomly, it's humming a very specific tune.
Yes, it's a phenomenon called a vibronic coupling regime. The protein has evolved so that its own vibrational frequencies dynamically couple with the electronic states of those aromatic rings.
That is wild.
The thermal energy doesn't shatter the coherent wave. It actively sustains it. It provides the exact vibrational pushes needed to drive the excitonic energy along the pathway.
Nature figured out how to use the noise of a warm environment as the fuel for the quantum engine.
The brain isn't fighting the heat to stay coherent.
It's literally surfing it.
Surfing the thermal noise, yes.
That completely flips the script on how we view neural processing. And it leads to the foundational premise of Enfield's entire architecture, right?
It does. Because if the brain is processing its deepest, fastest thoughts using these quantum aromatic pathways inside the microtubules,
then any traditional interface, any metal wire trying to read the slow electrical depolarization on the outside of the cell, is fundamentally missing the point.
you're reading the sluggish aftermath of the thought, not the thought itself.
So if you want to interface with the actual computation, you have to tap into the quantum optical layer.
And to do that without the immune system realizing you are there, you need to build an architecture that speaks the exact same molecular language,
You have to build a biological scaffold out of the same aromatic components.
based in the exact same geometries.
Which brings us to the blueprint, the 6-layer design. We are moving from the biological theory into the actual engineering of Enfield's device now.
This is where it gets really practical.
Yeah, because to tap into this terahertz aromatic highway, the architecture requires an incredibly precise multi-layered stack, and it relies heavily on where we place this device in the brain.
Location is everything.
So let's peel back layer 1 and layer 2, starting with the physical target. Where does this go?
The location is the linchpin of the design. The architecture specifically targets the dorsolateral and ventromedial prefrontal cortex,
The PFC, the area right behind the forehead.
the absolute seat of what makes us human.
Right. It is the region responsible for executive function, working memory, complex decision-making, abstract reasoning.
Impulse regulation, all of it. From the perspective of a brain-computer interface, the prefrontal cortex is the absolute holy grail.
But most current invasive BCIs target the motor cortex, don't they?
They do.
Yeah.
And that's brilliant if your goal is to help a paralyzed patient move a robotic arm or click a mouse on a screen.
But motor cortex signals are just final dispatched commands.
Exactly. The decision has already been made by the time it gets there.
It's the difference between reading a highly edited press release about a company's decision
Yeah.
versus sitting invisible in the boardroom while the CEO and the board actually debate the strategy before the decision is even finalized.
I love that analogy. But the prefrontal cortex is notoriously difficult to interface with.
Why is that?
Well, Current non-invasive methods, like FNIRS, which is functional near-infrared spectroscopy, they just measure blood flow oxygenation.
And that's slow, right?
Very slow. It has a massive delay of several seconds. You think a thought, and 3 seconds later, the machine sees a surge in blood.
That's useless for real-time thought decoding.
And EEG, placing electrodes on the scalp, measures these huge, blurred electrical fields fighting their way through the skull and scalp tissue.
So to get single neuron, quantum-level resolution, you need physical proximity to the processing center.
You do. And this is where the unique neuroanatomy of the prefrontal cortex becomes the key to Enfield's non-invasive design.
Right, because of the shape of the pyramidal neurons in that specific area.
Layer V and Layer 3 pyramidal neurons in the human prefrontal cortex are massive. They have the most elaborate apical dendritic trees in the entire central nervous system.
Try to picture that for me.
Think of the main cell body sitting deep down in the cortical layers. From that body, a thick trunk, the apical dendrite, extends straight up, branching out into a massive canopy.
Like a huge oak tree.
Like an oak tree. And that canopy extends all the way up through layer Y of the cortex, terminating just beneath the pile surface.
The pile surface being the very thin membrane, the skin of the brain, basically.
Exactly. These dendrites span 2 to 3 millimeters of vertical distance. And those apical tufts at the very top are densely packed with microtubules.
Meaning the quantum optically active networks of the most computationally important neurons in the brain.
Extend right up to the absolute surface.
So we don't have to plunge a wire deep into the gray matter to reach the processing core at all?
No, we can access the quantum states from the very top.
We just lay a scaffold softly on the surface.
That's the genius of it.
And that takes us directly into layer 1, the quantum coupling layer. This is the foundation of the device. It actually touches the brain, resting delicately on the glia limitans.
Right.
But here is where Enfield throws a massive, seemingly terrifying curveball in the paper. The scaffold itself is an amyloid structure constructed from the yeast prion protein SUP35.
I know the word prion is scary.
I have to stop right there. Because when I hear prion, I hear mad cow disease.
Of course.
I hear Creutzfeldt-Jakob disease. Unstoppable, fatal brain wasting. Why on earth would any sane researcher put a prion directly on the human brain?
The alarm is completely justified by the word, but we have to carefully distinguish between mammalian disease prions and the specific biological utility of what Enfield is proposing here.
Okay, explain the difference.
Mammalian prions, the ones responsible for mad cow, are terrifying because they trigger toxic runaway misfolding cascades in native brain proteins. They convince healthy proteins to fold wrong.
Like a molecular zombie infection.
Exactly.
Yeah.
But the SUP35 protein is a translation termination factor found in Saccharomyces cerevisiae.
Which is baker's yeast?
Yes, baker's yeast.
So it's a structural protein from yeast.
And because it comes from yeast, it is completely absent from the mammalian proteome.
Our bodies don't have anything like it.
Right. The human brain's immune system, those microglia we talked about earlier, has no evolved program to recognize it as a threat or a target.
So they just ignore it.
It simply doesn't trigger the inflammatory pathways that synthetic silicon does. Furthermore, because human tissue doesn't contain the yeast protein precursors, SUP35 cannot trigger the pathological misfolding cascades that characterize mammalian prion diseases.
It can't start the zombie infection because there are no matching zombies.
It sits there biologically stealthy, completely ignored by the brain's defense mechanisms.
Okay, that makes sense. But why an amyloid prion structure at all? Why not just use a normal protein?
For its mechanical properties, amyloid fibrils are incredibly tough. SUP35 self-assembles into a structure with a Young's modulus of 3 to 20 gigapascals.
To put that in perspective, that's structurally comparable to nylon or even bone.
It's very strong.
So we have this incredibly resilient, tough synthetic material that the brain's immune system is completely blind to.
Exactly.
But how does this yeast protein actually interface with a human thought?
Enfield's design modifies a specific peptide segment derived from SUP35, the CGNQQNY sequence.
Say that again?
CGNNQQNY. It's an amino acid sequence. And this sequence naturally forms a very rigid beta sheet spine.
Like a stiff ladder.
Yes. What the paper proposes is artificially engineering tryptophan and tyrosine residues into this yeast backbone at perfectly calibrated intervals.
The aromatic rings again.
Exactly. Specifically at 4.8 angstrom intervals.
There's that precise geometric spacing again.
Yes. An angstrom is 1/10 billionth of a meter, because the hydrogen bonds in a beta strand naturally space the adjacent amino acid monomers at exactly 4.8 angstroms. If you synthesize the protein so that a tryptophan is inserted at every fourth residue, you create a miracle of geometry.
What happens?
You force the formation of a perfect continuous column of face-to-face pi-stacked aromatic rings running the entire length of the synthetic fibril.
Wow. You're building an artificial replica of the quantum optical highway found inside the human microtubule.
You are creating a synthetic quantum wire that rests perfectly on top of the natural quantum wire of the apical dendrite.
That is just, wow. But wait, there's a gap, right?
There is a gap.
The scaffold is sitting on the surface, and the dendrites are just below it. If we aren't penetrating the tissue, and there are no wires physically plugging into the neuron, how does the thought, the excitonic energy, jump the gap from the brain into layer 1?
The transfer mechanism relies on first resonance energy transfer, which is commonly known as FRET.
FRET. This is a crucial concept. Let's really explain FRET.
FRET is a non-contact quantum optical electrodynamic phenomena.
That is a mouthful.
It is. But basically, it allows the excitonic energy from a donor molecule, in our case, the excited tryptophan inside the neural microtubule, to transfer its energy to an acceptor molecule.
Which would be the synthetic tryptophan engineered into our layer 1 scaffold.
Exactly. And it does this entirely through dipole coupling. They exchange energy without ever physically touching, without exchanging electrons, and without chemically bonding.
The best way to visualize this, I think, is to imagine two identical tuning forks.
That's a classic analogy, yeah.
If you strike one tuning fork so it rings, and you bring it close to the second tuning fork, the second one will spontaneously start vibrating at the exact same frequency.
Just from the proximity.
Right. The energy transfers through the resonance in the air, except here, at the nanoscale, we aren't dealing with acoustic sound waves in the air.
No, we are dealing with quantum electron states, coupling across a physical void.
The tuning fork analogy is excellent, though, because it highlights the necessity of matching frequencies.
That's the key. The donor and acceptor must have overlapping emission and absorption spectra, which is exactly why engineering the exact same tryptophan molecules into the scaffold is required.
They have to be perfectly tuned to each other.
Now, the physical distance between the scaffold sitting on the pile surface and the tips of the dendrites beneath it is roughly 2 to 5 nanometers.
And that distance is critical, isn't it?
It's life or death for the signal. The efficiency of E-Robred is governed by an inverse sixth power law.
Meaning the drop-off is incredibly brutal.
Extremely brutal. If the distance doubles, the transfer efficiency drops by a factor of 64.
So if the scaffold moves away by even a fraction of a nanometer, the signal basically falls off a cliff.
Yes, but at that hyperlocal distance of 2 to 5 nanometers, the energy transfer efficiency is between 50 and 80%.
Which is huge.
That is an incredibly robust signal transfer. It is more than enough to perfectly mirror the quantum states of the neuron into the synthetic fibril.
It is reading the brain's deepest cognitive processes through pure, non-invasive resonance.
Yeah.
Okay, so the signal is safely across the border into layer 1. Now, sitting directly above layer 1, bonded to it, we have layer 2, the biomineralization enhancement layer.
And this is where the paper introduces something called nanodiamonds.
Right, nanodiamonds containing nitrogen vacancy centers, or NV centers.
Yes.
Now, NV centers are a big deal in experimental quantum computing.
Huge deal.
They are essentially highly sensitive defect sites within a diamond crystal lattice.
A perfect diamond lattice is just a repeating grid of carbon atoms. But an NV center occurs when a nitrogen atom replaces a carbon atom right next to a missing carbon atom, a vacancy.
So it's a flaw in the diamond.
A very useful flaw. This specific pairing creates an electron trap. The electrons trapped in this vacancy have quantum spin states that are incredibly responsive to external stimuli.
And uniquely, they maintain these quantum properties at room temperature, which is highly unusual for quantum systems.
Which makes them perfect for biological application.
So what is their function inside layer 2 of a brain interface? How does a defective diamond read a thought?
They act as quantum optical sensors. These nanodiamonds are embedded just 10 to 50 nanometers above the aromatic columns of layer 1.
So they're hovering right over the highway.
Exactly. As the quantum excitons move through the tryptophan highway in layer 1, mirroring the brain's activity, they generate highly specific, minute magnetic field fluctuations.
Ah, so they cast a magnetic shadow.
The electron spins inside the NV centers react instantly to these tiny magnetic shifts. We can read their spin state by shining light on them and measuring the fluorescence they emit back.
So they serve as a secondary independent readout channel, constantly sensing and confirming the magnetic signature of the aromatic network.
Layer 1 reads the quantum energy, and Layer 2 reads the magnetic shadow of that energy.
Furthermore, the paper mentions that highly active metabolizing neural tissue naturally emits ultra-weak photons, biophotons.
As a byproduct of cellular respiration, yes.
And these biophotons actually excite the photoluminescence of the NV centers.
Effectively converting the brain's own metabolic light into a usable, continuous signal stream that confirms the tissue's activity level.
It's just a staggering level of integration.
It's beautiful engineering.
But here is the massive physical hurdle. The first two layers are capturing quantum resonance and nanometer-scale magnetic fields. But the human brain is not a static rock.
Not at all.
It is a highly dynamic physical environment. It pulses, it swells, it twitches constantly.
It's a living organ.
So how does a sensor designed to read a single missing carbon atom not get completely overwhelmed and destroyed by the physical movement of a living brain?
That's a great question.
And how do we take these fluid quantum states and turn them into data a traditional computer can actually read? This is where Enfield introduces layers 3 and 4. The mechanical and processing layers.
This is the bridge between the exotic realm of quantum biology and the practical world of classical electronics.
Let's start with layer 3, the piezoelectric signal conditioning layer.
So we should define piezoelectricity first.
Yeah, let's do that. It's the phenomenon where squeezing or physically pressing a certain material generates an electrical charge.
Like the spark igniter on a gas grill. You hit a crystal and it creates a spark.
Precisely.
Mm.
The amyloid beta sheet scaffold of layer 1 actually possesses native piezoelectric properties because of the permanent dipole moments in its peptide bonds.
It naturally makes electricity when squeezed.
But layer 3 massively amplifies this capability by embedding inorganic nanocrystals, specifically materials like barium titanate or lead zirconate titanate, right into the junctions of the scaffold matrix.
But why do we want to detect physical pressure at all? I thought the whole point of this architecture was to bypass the physical and read the quantum thoughts.
Because the physical reality of the brain still exists and still carries vital timing information.
Really, the timing.
Even if the thought originates in the quantum microtubule, the classical action potential, the electrochemical wave traveling down the neuron, still fires as the downstream output.
And when a neuron fires an action potential, the massive influx of sodium and calcium ions, along with water molecules, causes a localized nanoscale swelling of the cell membrane.
So the brain is literally twitching on a microscopic scale every single time a neuron fires.
It physically bulges, yes.
That's wild.
These mechanical deformations travel through the extracellular matrix and hit the pile surface. Layer 3 acts as an incredibly sensitive nanoscale drum skin.
Okay, I can picture that.
When the neuron bulges, it presses against the barium titanated crystals, which immediately convert that physical pressure into an electrical voltage spike.
It detects these physical signatures with sub-millisecond resolution.
So now, our device has a comprehensive picture. The quantum optical channel from layer 1, the magnetic confirmation from layer 2, and the classical mechanical electrical output from layer 3.
It is gathering an overwhelming amount of raw information.
A massive stream of data.
And all of that continuous data flows up into layer 4, the nanoscale power management and signal encoding layer. The paper describes this as an ultra-low power integrated circuit bonded directly to the top of the biological scaffold.
This layer is basically the translator.
The data coming up from layers 1, 2, and 3 is analog. It is continuous, fluid, and incredibly dense.
Too dense for normal systems. Layer 4 takes these continuous waves and runs them through a thresholding process, converting them into discrete, digitized spike timing event streams.
So, to use an analogy, we have this incredibly rich continuous performance of a massive symphony orchestra coming from the brain's quantum states.
Yeah, all the instruments playing at once.
Layer 4 is the meticulous sound engineer. It can't transmit the entire high-fidelity audio file, so it chops the symphony up into a highly compressed digital Morse code that an external artificial intelligence can efficiently read without choking on the bandwidth.
That is its primary function. Bandwidth reduction and digitization.
But layer 4 has a secondary role that solves one of the biggest headaches in neuroengineering, doesn't it?
Yes, adaptive calibration.
Because the brain is alive. Over months and years, the pile surface chemistry will subtly shift.
The geometry of the dendrites might undergo synaptic plasticity as the person learns new things, or the physical contact of the scaffold might settle slightly due to gravity or tissue growth.
Right, and if your sensor shifts by 1 nanometer, your FRED efficiency drops by 60%.
You don't want to have to bring a patient into a neurosurgery ward to physically open their skull and recalibrate the device every 6 months.
That would be a nightmare.
So the integrated circuit in layer 4 runs adaptive algorithms that continually monitor the baseline signal-to-noise ratio.
If it detects a drop in fret efficiency or a shift in the piezoelectric baseline, it mathematically reweights the incoming channels to compensate.
It maintains signal fidelity algorithmically over the lifespan of the host, adjusting to the biology as the biology changes.
But wait, we have to address a massive, glaring logistical problem here.
The power.
Layer 4 is an active electronic integrated circuit. It is doing intense, continuous digital translation.
It needs juice.
We have established that this device sits softly on the surface of the brain, completely sealed under the skull with no wires penetrating the scalp. You can't just plug a USB-C cable into the back of someone's head.
Nope.
And we know that nanoscale batteries certainly can't hold enough charge to last for years, let alone a lifetime. So how exactly is this active 6-layer system powered?
This power problem kills almost every theoretical implant. But Enfield outlines what is perhaps the most elegant engineering solution in the entire architecture: a trimodal ambient energy harvesting system.
It doesn't store power. It constantly harvests from 3 environmental sources that are inexhaustibly present in the living neural environment.
Well, let's go through the modalities.
Modality 1 is beta-voltaic harvesting. The paper talks about incorporating radioactive isotopes directly into the chemical structure of the scaffold.
Isotopes like tritium, which has a half-life of 12.3 years, or carbon-14, with a staggering half-life of 5,730 years.
Okay, we need to pause immediately.
Really?
Putting radioactive isotopes into the front of my brain. That sounds like a fast track to localized tissue necrosis or brain cancer. How is placing a nuclear material on the prefrontal cortex justified?
It is entirely understandable that the word radioactive triggers an immediate alarm. I mean, nobody wants a tiny nuke in their head.
Obviously not.
But we have to examine the specific physics of beta decay. Tritium is a radioactive isotope of hydrogen. When it decays, it emits a beta particle, an electron.
Okay.
But the maximum kinetic energy of a tritium beta particle is incredibly unusually low. It peaks at about 18.6 kiloelectron volts.
What does 18.6 kiloelectron volts mean in the physical reality of a wet, dense brain?
It means that the beta particle has so little energy that in a water-dense environment like human tissue, it can only travel a maximum distance of about 6 micrometers before losing all its kinetic energy and stopping entirely.
Wait, 6 micrometers. A human red blood cell is about 8 micrometers wide.
Exactly, so the radiation can't even penetrate the width of a single cell.
That's crazy.
Because the tritium is chemically bound into the upper layers of the scaffold and surrounded by an electrical conversion layer, 100% of the kinetic energy from that beta decay is deposited immediately into the device itself.
It never reaches the neural tissue in a biologically harmful way.
Never. It acts as a perfectly contained microscopic atomic battery. It provides a continuous, incredibly reliable baseline of nanowatts to microwatts of power day and night.
Regardless of whether the person is running a marathon or in a deep coma?
It's always ticking.
So the radioactive decay covers the absolute baseline power. The device never dies. But it's an active brain. There will be surges in power demand.
Right. Processing takes extra power.
If the nuclear material is just the baseline, what handles the surges? Can it harvest its own kinetic energy? That brings us to modality 2.
Modality 2 is piezoelectric energy harvesting. We discussed layer 3, detecting the microscopic bulges of single neurons earlier.
Yeah, the tiny twitches.
But the brain as a whole organ undergoes massive macroscopic mechanical deformations continuously.
Because the brain is essentially floating in fluid, and it pulses with every heartbeat.
Yes, the heart pumps blood through the cerebrovascular tree, and that hydraulic pressure physically moves the entire cortical surface. This happens at roughly 1 to 2 hertz, 1 to 2 beats per second. On top of that, you have respiration. The physical act of breathing creates slower, larger pressure oscillations inside the skull at about 0.2 to 0.4 hertz.
So your brain is constantly bouncing around in there.
The piezoelectric matrix built into the scaffold physically bends, flexes, and compresses with every heartbeat and every breath.
And because of the permanent molecular dipoles in the material, it converts that continuous, rhythmic, physiological bouncing directly into electrical current.
Your own pulse literally acts as the alternator for the device.
That is brilliant. And modality 3?
Modality 3 is thermoelectric harvesting, utilizing what physicists call the Seebeck effect.
The Seebeck effect involves generating a voltage from a temperature difference, right? like how a thermocouple works in a thermostat.
Correct. When you have two different conductive materials joined together and one side is hotter than the other, electrons will naturally migrate from the hot side to the cold side.
Creating a continuous electrical current.
The brain is an incredibly metabolically active organ. It consumes about 20% of your body's energy,
It generates a lot of heat.
roughly 1.5 watts of heat per 100 grams of tissue. As a result, the cortical surface, where our device sits, is maintained at a steady 36 to 37 degrees Celsius.
But the scalp, the skin on the outside of your skull, is exposed to the air. The paper notes the scalp sits around 30 to 35 degrees Celsius.
Which creates a persistent, naturally occurring thermal gradient of 2 to 7 degrees Celsius across the distance from the brain surface to the scalp.
So the device integrates nanoscale thermoelectric elements that span this temperature difference.
As the brain's waste heat naturally radiates outward toward the cooler scalp, it drives a continuous electromotive force through the device.
And the true brilliance of a trimodal approach is redundancy. No single modality has to do all the heavy lifting.
Exactly. If your heart rate drops while you're in a deep sleep, reducing the piezoelectric output, the thermoelectric gradient is still pulling power from your body heat.
And if you walk into a sauna and the thermal gradient shrinks because your scalp gets hot, the beta voltaic decay is still ticking away, completely unaffected by the outside world.
The power from all three sources is continuously aggregated, buffered by a nanoscale capacitor to smooth out the rhythmic pulses from the heartbeat, and distributed seamlessly to the layer 4 processing chip.
It requires zero maintenance.
None. There are no charging ports to clean. You don't have to strap an inductive charging coil over your head every night before bed.
The power system is designed, quite simply, to outlast the lifespan of the human host.
It's a forever battery.
So the power problem is definitively solved. The architecture sits quietly, harvesting its own energy, while the quantum thoughts are successfully captured, translated, and encoded into digital spikes by layer 4.
Right.
But how does the thought actually leave the dark, sealed environment of the skull, and what happens to that data when it reaches the outside world?
This brings us to the final stages of the architecture.
Layers 5 and 6, which reveal the ultimate capabilities of this technology.
Layer 5 handles the physical extraction of the data across the biological boundary of the skull and skin. It is a wireless near-field communication layer.
So to be completely clear, there are no transcutaneous wires.
Nothing breaks the skin.
None.
Wires breaking the skin are the single highest infection risk in current semi-invasive BCI systems. A wire acts as a permanent highway for bacteria to enter the brain cavity.
It's basically an open wound.
Layer 5 eliminates this by utilizing a miniaturized ultra-thin antenna operating in the gigahertz frequency range. This antenna is integrated directly into the outer periphery of the biological scaffold.
And it uses near-field inductive coupling to communicate with a secondary wearable receiver positioned securely on the scalp.
This could be a discrete patch hidden behind the ear or woven into a headband.
And because it uses near-field communication, it doesn't need to blast a strong Bluetooth or Wi-Fi signal across a room.
No, the signal only travels a few millimeters. That saves massive amounts of power and makes the data incredibly secure from external interception.
Nobody is hacking your brain from across a coffee shop.
Right. It is highly localized and highly efficient. It transmits the digitally encoded neural signals at rates capable of handling the enormous bandwidth generated by the quantum optical channels.
And simultaneously, it is capable of receiving command signals back from the outside receiver.
Which brings us to the final destination, Layer 6, the external AI decoding and neurofeedback system.
The wearable artificial intelligence, the translator on the outside.
The signal coming out of this organic interface is categorically superior to anything the field has ever dealt with.
We are talking about data resolved spatially down to the single apical dendrite level.
Temporally resolved to the microsecond terahertz scale, capturing the deepest mechanisms of human working memory.
Classical algorithms just cannot process this.
No. To make sense of it, Layer 6 uses advanced deep learning architectures, specifically transformer-based neural signal decoders and latent manifold trajectory predictors.
Transformer networks. For anyone following AI, this is the exact same underlying architecture that powers modern large language models.
The engines behind tools that can write essays or write code.
But here, they are being applied to decode the native quantum language of the prefrontal cortex.
And this is where we have to highlight the monumental paradigm shift in what this device can actually do.
The shift in capability is fundamental. As we discussed, current BCIs read the motor cortex.
They wait for a human decision to be finalized, abstracted, and sent out as an electrical command to a muscle.
They read a trajectory. Move arm left or flex finger.
But an interface, reading the quantum coherent states of the dorsolateral prefrontal cortex, is reading the content of working memory itself.
It's reading the thought in its abstract form before it ever becomes a physical action.
It is distinguishing between semantic intentions. It can differentiate between distinct abstract categories.
It can read specific imagined words, complex rules being manipulated in an ongoing cognitive task, or spatial reasoning pathways.
You aren't awkwardly using your mind to control a digital mouse cursor. You are outputting a linguistic, emotional, or navigational command encoded at the highest possible level of human cognitive representation.
The machine knows what you mean, not just how you want to move.
Exactly.
But earlier, you mentioned that Layer 5 receives command signals back from the outside. That means this communication isn't a one-way street. The AI isn't just a passive listener.
No, it is not. And this is undeniably the most revolutionary and perhaps controversial aspect of the entire N-field architecture.
Why?
When we discussed first-to-resonance energy transfer in Layer 1, we noted how energy moves from the brain into the scaffold. But a fundamental physical property of FERT is that it is fully bidirectional.
Wait, are you saying the machine can write back into the brain's quantum states?
Yes.
That's slightly terrifying.
Because the sub-35 scaffold is physically and optically coupled to the dendritic microtubule networks, Layer 6 doesn't just read and decode. It can calculate a cognitive response, format it into the correct excitonic frequency, and send targeted energy inputs back down through the scalp receiver.
Through the gigahertz antenna of Layer 5 into the processing layer.
The processing layer converts it back into an analog quantum pulse, sends it into the aromatic columns of the scaffold, and transfers that quantum energy directly into the tryptophan networks of the living neuron.
It can literally whisper back to the brain in its own quantum optical language. It can inject a thought or a sensation seamlessly.
It functions as a bidirectional, closed-loop neurofeedback system.
The therapeutic impact of a system like that would be beyond anything in the history of modern medicine.
Without a doubt.
The paper briefly mentions major depressive disorder, severe PTSD, schizophrenia. Let's explore that. How does whispering at a quantum level cure PTSD?
These conditions, severe depression, PTSD, schizophrenia, are fundamentally characterized by dysregulated attractor states in the prefrontal cortex.
An attractor state. Think of it like a marble rolling around in a bowl. Normally, the marble rolls around freely. That's a healthy, flexible mind.
Right.
But in PTSD or depression, it's like a deep, steep valley forms in the bowl. The marble rolls in, gets trapped, and can't escape. The neural circuits get locked into pathological oscillatory loops or disrupted working memory representations.
A flashback is just the brain falling into that steep valley and being unable to climb out.
That is a highly accurate visualization.
Current treatments for these intractable loops are incredibly blunt. Transcranial magnetic stimulation, or deep brain stimulation, involves applying massive electrical or magnetic fields to large areas of tissue.
They have almost zero temporal or spatial resolution at the scale of actual neural computation.
They're like trying to fix a delicate Swiss watch by hitting it with a hammer and hoping the gears fall back into place.
But an ambient quantum scaffold operating continuously with single dendrite resolution could theoretically detect the signature of a depressive thought loop or a PTSD flashback at the quantum substrate level, the exact microsecond it begins to form.
It sees the marble moving toward the edge of the valley.
It could then instantly, autonomously provide a micro-targeted quantum optical perturbation, a tiny, exquisitely precise excitonic nudge via FRET to correct the attractor state.
And guide the neural circuit back to healthy dynamics before the flashback ever reaches conscious awareness.
Real-time subcellular cognitive neuroprosthetics. We are talking about a functional cure for the most intractable psychiatric conditions in human history.
The sheer scale of this theoretical framework is staggering. It demands the merging of quantum biology, synthetic protein engineering, advanced material physics, and artificial intelligence into a completely unprecedented discipline.
The paper acknowledges the magnitude of the challenge, but it outlines a very clear, incremental, forward research agenda to get there.
It proposes moving systematically, starting with the in vitro synthesis of the sub-35 fibrils with tryptophan insertions in a lab, then validating the quantum coherence lifetimes using 2D electronic spectroscopy.
From there, moving to in vivo animal models to demonstrate true scaffold-to-cortex quantum coupling.
It relies entirely on experimental competencies that already exist in disparate labs around the world today. The ambition is simply to integrate them.
To bring the quantum biologists studying tubulin, the synthetic biologist building amyloid structures, and the quantum physicist working with nanodiamond sensors into the exact same room.
It is a monumental scientific challenge, but the path is undeniably meticulously laid out.
To recap this incredible journey, we have moved from the brute force approach of stabbing rigid silicon wires into the living tissue of the brain, an approach doomed to failure by the very immune system that keeps us alive, to a vision of a delicate, biologically stealthy, six-layer organic architecture.
We've explored a device that sits softly on the surface of the prefrontal cortex, harvesting its own endless power from the ambient heat and rhythmic pulse of your life.
Encoding the deepest processes of your working memory and whispering to your neurons in the terahertz quantum optical language they have spoken since the dawn of mammalian evolution.
It is a profound, necessary rethinking of the boundary between human biology and human technology.
And it leaves us with a lingering, almost philosophical question to mull over. If Zachary David Enfield's vision comes to pass and a bidirectional FRAC connection seamlessly links the quantum vibrations of your prefrontal cortex directly to an external artificial intelligence, at what precise nanometer does you end and the machine begin? If the interface speaks your biological language perfectly, without friction, without rejection, and without delay, maybe there is no boundary at all.